Okay, so everybody, I am really sorry about Monday. It was my mistake, kind of kicking myself when I arrived because, well, I'll explain to you. So, like, I made a mistake when I copied over my timetable into my Google calendar. So I'd put the class here, right? And then, even though I did check on the online timetable, which is this one. Where are we? Yeah, I didn't realize that it was put like at 2 p.m. On Monday. I just saw, oh, it's after lunch. Okay, so I went back after lunch after my first class and then came back here at 3:30. So I was kind of feeling a bit stupid about that. I'm really sorry. So whoever has my phone number, if ever like I don't turn up another time, try and contacting me, please. But I assure you it will never happen again. Definitely never. Okay, so anyway, so thanks for all coming this afternoon. Right, so a little bit of an update on Microbe of the Week. So the question was, what is so special? What kind of carbon source is used by a bacterium called Methylo-2V Microbium burriatensi? And so what's the carbon source? What's so special about this one compared to the other bacteria of this type? And when was this genus name invented? Because when I read this, I had certainly had never heard of Methylo-2V microbium genus before. So the answers were it uses methane as its own sole carbon source. It's a methanotroph. The thing that's important about it, why it could be useful to try and mitigate global warming, is that it can use methane when it's present in relatively low concentrations, like less than 500 parts per million. Now that is quite unusual for methanotrophs because most of these bacteria grow only when there is a high concentration of methane. And in places where it would be useful to remove methane, or at least convert it to carbon dioxide, then the methane concentration is much lower. So Methylo-2V microbium, burriatensi, could actually be useful in a real-life situation, unlike other methanotrophs. So that's what's interesting about this. And there was a paper published on this last August and had a bit of media coverage. The other question was, so when was the genus name invented? 2018. There was a paper in 2018 where people tried to, well, the researchers wanted to look at the phylogeny of methanotrophs. So they sequenced a bunch of genes and reorganized the phylogeny of methanotrophs, and they found that this one, burriatensi, and a few other strains formed a distinct clade, and they proposed a new genus name for this group of microorganisms. So thanks to everybody who sent answers in. Once again, some quite nice answers. And here we've got the leaderboard so far. So Solène, Kiridwen, and Guy Alec are the ones in the lead here. So there's the ones to catch. I don't plan on doing another one this evening. You want to do a Microbe of the Week this evening? No? Okay, right, yeah. Me neither. That's okay. Right, so one of the things I left in suspense last time was, I asked you to have a look and see how could you tell if bacteria are VBNC, viable but non-culturable, or really dead, because if you can't cultivate them, they won't grow on an agar plate. So it's difficult to tell if they are really alive. So anybody have any ideas for this, just to like open it up? Yeah? Yeah, I think that's the real test, isn't it? So the answer here is you put the bacteria back into a very rich medium and see if you can get them to resuscitate, and that's really the proof that they can actually come back to life. So yeah, that's the main thing. But of course, this might take some time, right? Because you don't know how long it will take for them to reactivate, if it's possible. And one of the other issues with that approach is you might not know what the optimal stimulus would be to, reactivate them. So if you get a negative result, you'll still be kind of wondering, are they really dead, or did I just not use the right conditions? So it's kind of difficult. So yes, it's true, but there are other approaches to differentiate live and dead bacteria. Let me give you a clue here. Which one of these is alive? This one or that one? This one is alive. Yeah, this is supposed to be the plasma membrane. And this thing, if it's got holes in the plasma membrane, this will be dead. So, which one of these can be stained with a non-membrane permeable dye? This one, right? So you can get some kind of dyes that will only stain DNA, for example, if the membrane is not intact. So VB and C bacteria will have an intact membrane. Okay, whereas dead bacteria, their plasma membrane will be disrupted. The other thing that you can also do is try and detect some basal metabolic activity. So for example, in a dead cell, you won't find any ATP, but in a live cell, even if it's like dormant, you should find some ATP present. So you can try and distinguish cells that have no biochemical activity to cells that still maintain some low-level biochemical activity. And this you can do on a suspension of cells and get an answer pretty quickly. Okay. So you've got a bunch of biochemical tests to see whether VBNC is really different from live cell, from dead cells, but in the end you have to be able to resuscitate them somehow. Okay, so we saw this. Don't think we saw this, right? We stopped at the end of this slide. Is that right? Yeah. So let's just want everybody to kind of think about these equations here. And just to make sure you understood this correctly, I would like you to have a think and send in an answer to this question. Yeah. 16 votes here. 17. Okay, this is what you think. So nobody thinks this is a trick question. Okay, and you're right, it's not a trick question. There is one piece of information lacking. No, you don't need to know what it is, okay? So for any bacterium in suspension, there will be a relationship, a linear relationship between the OD600 and the number of CFU per ml, colony forming units per ml. So that will be different depending on the bacterium, but the relationship will be linear. So you don't really need to know 4. So at least 1 or 2. Now the answer is not number 1, but I understand why a lot of people think this, and I noticed that that's what people think because in the lab class, often when you do this and you make this measurement, and often what a lot of people will do is they'll get their OD600 measurement, convert it into bacterial CFU per ml, and then do the calculation on the CFU per ml. But you don't really need to do that because if you remember a few slides ago, I said there's a linear relationship between the OD600 and the number of CFU per ml. So that means that if you rearrange this, that your OD600 divided by K, that will give you CFU per ml, okay? Now, think about this equation that you're going to use to, why am I not, it's bizarre, I should be doing a slideshow here. So, if you're going to perform this calculation, what you want to do is this, right? You're going to use this relationship. So, your number of cells at the time t is you'll get it from like this, okay? And I'm going to take the equation from here, right? So imagine that you're converting your OD600 into CFU per ml. So you've got n equals the log of your OD600 at time t divided by k minus the log of OD600 at time 0 divided by the same constant. And all of that divided by log 2. So basically this is going to be the OD600 at time t, or the log of this minus the log of k. And this is going to be the log of this minus the log of k. So these ones are going to disappear. Right? Because that's the way logarithms work. So basically, everybody got this? Yeah, and I'm saying the k comes from here because I said there's a linear relationship between the OD at 600, the optical density at 600 nanometers, and the number of colony forming units in a suspension, the number of bacteria. So if it's a linear relationship, that means that this optical density equals k times the number of bacteria, a constant, okay? And then you can rearrange this and say that the OD600 divided by k, that gives me my CFU per ml. Okay? But the thing is, later on in your equation, the constant is going to disappear. So what this means is that Oh, when we get back to the original question, it means that you do not need to know what K is, okay? Because later on in this equation, it's just going to disappear. So you can perform the equation just with the OD 600 measurements directly. Okay, so I'm spending a bit of time on this now because afterwards in the lab class, everybody will do this. And in fact, it's easier than you think, right? Because you just put in the OD 600 measurements and it works. So this is not the correct answer, which means that answer 2 is correct. Now, why is this correct? Because in the question, all you know is you've got at time 0, well, your time 0, whatever it is, you have OD 600 is 0.1, and at some other time point, your OD 600 is 0.5. And the problem is, and you're saying, what I want to do is imagine that this is, and also don't forget you should have this on a log scale. And you should say that on a log scale, this should be linear. Okay, then I can work out, then my equation is going to work. But in fact, with the information here, you don't really know whether you're in the exponential phase because the real growth curve might be like this. Okay, from here to there, bacteria might be in the stationary phase. And in which case, you're going to calculate this is your growth rate, but in reality, it's this fast. But also, the real thing, real growth curve, could be like this. And this will also be wrong if you calculate that you are just going from this point to that point. So the lesson here is what? The lesson here is just with 2 data points, generally it's not enough. Unless you have in the description that you know that this is in the exponential phase. So this is why when you do this experimentally, you have to take several time points so that you can tell, so that you know that you're in the exponential phase of growth. Okay, so these 2 things, very important when you're actually doing this calculation. You can use the OD 600 measurement directly, but you need to know that you're in the exponential phase. Okay, so up to now we've talked about cultures in a closed system, okay, where you inoculate the bacteria into a certain volume of culture medium, they grow up to a maximum, they reach a stationary phase, and then they begin to die. But there is another way to try and grow bacteria in what is called a continuous culture. So in this kind of system, bacteria are inoculated, and then they're maintained in the exponential phase by continually adding new medium and taking out the excess of the bacterial culture. So you set up something, and this kind of equipment is called a chemostat because the chemical environment is static. So it kind of looks like this. So this is your culture vessel here, and you've got an input of air, and you've got something to turn around like a propeller and keep the bacteria in suspension. And you'll have some kind of constant flow of fresh medium comes into this culture vessel. So they always have fresh medium to eat. And then to keep it constant, you've got to like take away the excess of the culture and put it into some collecting vessel. And this is what you're going to harvest at the end of your experiment. Now, the thing about this is you have to be careful with the flow rate, because if the flow rate is not enough, then the bacteria will still be in the, get to the stationary phase. But if the flow rate is too fast, then you wash out all the bacteria and you just have like fresh medium here and no bacteria. So that's kind of the problem, more a chemical engineering problem than a microbiology problem, I have to admit. So basically the whole thing about the chemostat is how to work out the optimal dilution rate. So the dilution rate is the flow rate divided by the chemostat volume. So if I have a flow rate of 1 or a dilution rate of 1, that means that all of the volume of the chemostat is replaced in 1 hour. Or it can be 2, it can be 5, whatever. It can be higher, greater than 1. But at some point, so the idea is that when you add the fresh medium, whatever the limiting nutrient is, its concentration will go up, and that will allow more bacterial growth. So you'll get to a kind of like, if you keep adding new medium, then your bacterial growth can get up to its maximum. But at some point, if your flow rate increases too much, then more bacteria are going to be washed out of the culture. And in fact, what will happen is the nutrients aren't used up anymore, so the nutrient concentration goes way too high, but the bacteria are washed out of the vessel, so it comes down to 0. Now, if you're running a chemostat, you want it to be working around this zone. And, it's weirdly, I know why this happened. It's annoying, but okay, so the setup is something like this in that case. So what you've got to do is you've got to put an OD600 detector here at the output, and this will control the pump that will adjust the flow rate. So if what's going to happen is you keep increasing the flow rate as long as the OD600 measurement here is increasing, but as you increase the flow rate, you'll get a maximum, and then if you increase the flow rate even more, once this starts going down, then you've got to control the pump to reduce the flow rate, to make sure you stay in this zone. So, that's what a chemostat is, and it has some uses in industry. Okay. So in industry, it's used in biotechnology to maximize, the yield of the bacterial product, and can be used in research if you want to study bacteria in the exponential growth phase over a long period of time. Okay, so that's bacterial culture in liquid, either in a closed system or in a chemostat. But a lot of bacteria, most of the time, actually grow on surfaces. And when they grow on surfaces, they grow in the form of a biofilm. And a biofilm is a microbial community composed of one or several microbes in an organic matrix that they produce themselves. So basically, any kind of film that accumulates over time. If you ever looked in the tubes underneath the sink in your kitchen, when they get blocked, it's kind of horrible black gunk. Okay, that's a microbial biofilm that's growing in there. And, some of these biofilms can be monospecies, just one bacterium and its organic matrix. So Pseudomonas aeruginosa in lung infections is very, very difficult to cough out of the lungs, especially people who have cystic fibrosis. It's a very, very dangerous infection for them. And the bacteria are difficult to eradicate because they grow in a biofilm inside the lungs. Staphylococcus aureus, again, medical problem. It can produce a biofilm and it coats medical implants and catheters. And these bacteria in the biofilm are difficult to treat because the biofilm is relatively impermeable to antibiotics. So they're real problems. Now, most biofilms out in a natural environment are composed of several species of bacteria, or sometimes with bacteria and and fungi as well. So if you've got this kind of grey film on your shower curtain, right, back at home, think about it, you have to keep cleaning it off, but it always comes back. That will be Sphingomonas and Methylobacterium forming a biofilm. Dental plaque that your dentist scrapes off your teeth, this is a biofilm. Okay, several species of bacteria all growing together in a kind of quasi-mineralized substance. And stromatolites, okay, like these assemblies of photosynthetic bacteria that grow in mats. So there will be not just one species of bacterium, but several growing in this biofilm. Now, biofilm growth has several phases, just as we saw like growth in a liquid culture. You've got a latent phase, exponential growth, stage 3 phase, and then death. You have different phases of growth of bacterial biofilms. So phase 1 is loose adhesion of the bacteria to a surface. And phase 2 is strong adhesion to the surface. Now, what's going to be necessary for this phase 2? Can anybody have a guess here? What kind of structure will bacteria need to adhere strongly to the surface? Yeah. Pili, yes. Okay. Adhesion is a function of the pili. So, firstly, they won't be expressing any of these adhesion pili, and secondly, they will start expressing them, and these are going to stick the bacteria to the surface. Now, the third phase is beginning to synthesize an exopolymer. So once you have enough bacteria locally present on the surface, then altogether they're going to start synthesizing some kind of biopolymer which they like to grow inside. And the fourth phase is maturation. So this kind of lump of bacteria growing inside this biopolymer that they've synthesized gets bigger and bigger. So, they're not just, few 100,000 bacteria flat on the surface here. They kind of like grow into a thick layer with more bacteria present. And then finally, you have dispersion. So also this is a controlled process. The bacteria will start to synthesize enzymes that break down the exopolymer and start to also synthesize what kind of structure that we can see here? Flagella, that's right, so they can start swimming around. So bacteria that form biofilms, often they alternate between what's called a pelagic phase, when they're swimming free in the liquid environment, and a biofilm phase. So it's a kind of cycle. So these bacteria that are swimming around, they're going to go and colonize a surface somewhere else. Okay? So you have to kind of think that whenever you're culturing bacteria in a liquid culture and shaking it, now you're getting something that's representative of the pelagic phase of bacterial growth, but it doesn't tell you everything about bacterial growth. This biofilm phase is very important. Now, in order for this to work, phase 3 here, the exopolymer synthesis, has got to be coordinated. All of the bacteria that are locally present have got to start synthesizing this polymer at the same time, because that's the only way that there can be enough of it to produce the biofilm. So that's a kind of interesting process, which is called quorum sensing. Now I know why this happened. And that means that bacteria are going to be behaving in a coordinated way. So this was first identified in Vibrio fischeri, which is a symbiotic bacterium in the kind of light-producing organs of a squid. And these bacteria produce an enzyme called luciferase that generates light, but only if they are present at a very high concentration. So the bacteriologists who are working on Vibrio fischeri, removed from the octopus or the squid, the light organ, to try and see how they produce light. They noticed that they only produce light if the culture is at high density. And they tried to figure out, how do the bacteria know how many other bacteria are around? Okay, that's the question here. And it works because they produce what's called an auto-inducing molecule. So in Vibrio fischeri and in a lot of gram-negatives, this auto-inducing molecule is a small molecule called N-acylhomoserine lactone, AHL. And this small molecule is membrane permeable. So once it's produced inside the cell, it can diffuse outside the cell. And if it's outside of the cell at a large concentration, it can diffuse back into the bacteria. So AHL is produced by this enzyme called LuxI, for the luciferase inducer. So the AHL that's synthesized in the cytosol will diffuse out of the cell. Now, if you've got a small amount of bacteria present, the concentration outside will be very small, and it will diffuse away and nothing happens. But if bacteria grow to a high enough concentration, the AHL concentration outside the bacteria can grow up to about 1 micromolar. And at this point, enough is present for it to rediffuse back into the bacteria and combine with this gene regulator called LuxR, which is a transcription factor, which will, once it's bound to the AHL, it binds to the promoter of the Lux locus and induces expression of the luciferase, and it also induces the expression of LuxI. So you get a positive feedback loop. As soon as you've got enough concentration of AHL outside the bacterium, then you get auto-induction of its production. So once it gets past a threshold, then the concentration really, really increases fast. Now, it doesn't get completely out of control because the LuxR transcription factor is, well, is under negative control by itself. So if the concentration is really, really too high, the system shuts down. Okay, so this is the way that all bacteria, the bacteria can detect how many other bacteria of the same type are present locally. Because if you've got a lot of bacteria locally producing this thing, then the concentration will be high enough for it to start the cycle and induce the luciferase genes. So that's why it's called quorum sensing, because it allows a bacteria to sense or detect when there are enough other bacteria of the same strain present locally. Now, many bacteria have quorum sensing systems. So in gram-negatives, there are a lot of acylhomoserine lactone. So in agrobacterium tumefaciens, this controls plasmid transfer between bacteria. And in Pseudomonas aeruginosa, that we just spoke about, it controls the expression of virulence factors and biofilm formation. Now, in gram-positives, they use a different system. They use auto-inducing oligopeptides. So, for example, in Staphylococcus aureus, quorum sensing by auto-inducing peptides induces the expression of virulence genes. And in Bacillus subtilis, it induces sporulation. So that kind of makes sense, right? If your bacteria, if there's too many of you around, then you can't carry on growing anymore. So it's a good idea to start producing spores. So, damn it. Just so annoying. I know why this is happening as well. So auto-inducing peptides, how are they produced? Well, they are first produced by a gene that codes for the a protein, and then this protein is cut up to release the auto-inducing peptide, which is going to be exported outside of the bacterium. So it's a similar kind of thing. If you've just got one bacterium hanging around there, the concentration is going to be almost nothing outside the cell. If you have enough bacteria, the concentration increases, and then it can be detected by a transmembrane sensor, which is in the plasma membrane of the bacterium. And this receptor is associated with a kinase activity, which will phosphorylate a regulator, which will go and induce the expression of the target genes. So, why can't this system work in gram-negative bacteria? Auto-inducing peptides, why won't they work in gram-negative bacteria? To give you a clue, it's a bit like the vancomycin ampicillin question. Yeah.
Do you have more layers about that one?
Yeah, you're on the right track. Be a bit more specific. So my auto-inducing peptide can diffuse across the peptidoglycan layer because the mesh is not really that tight. Well, it's pretty tight, but small peptide can get across. And then you've got the receptor, the sensor kinase here, which is called H. You can detect it. What about in my gram-negative? What's going to be the problem? Yeah, can't pass through the outer membrane. So it's not membrane permeable, okay, and it's too big to go through the porins. So porins will let through a molecule like glucose, maltose, some small antibiotics, but a peptide is too big. One amino acid, okay, can get through. Peptide, too big. Okay, whereas the acyl homoserine lactone is membrane permeable. It just diffuses across this membrane, diffuses across the other one. Okay, and also acyl homoserine lactone, very small, can get across the porins and then diffuse across the plasma membrane. Okay, so I think, once again, bacterial cell wall structure explains a little bit why gram-positives and gram-negatives have to use different systems for quorum sensing. Okay, so this is kind of the end of the first part here. So just to go back to what we saw about trophic types, chemotroph, phototroph, organotroph, heterotroph, etc. One big thing here, never mix up auxotroph and autotroph. It's very easy to do because they are so similar. So autotroph can fix inorganic carbon, uses CO2. Auxotroph means that it lacks the ability to produce some particular growth factor. So this one, you could easily mix those up. Definition of use of different types of media, growth curve. You have to know how to perform doubling time calculation. And quorum cell sensing, very important for producing biofilms. Okay, now we have a little bit less than 1 hour, 40 minutes, I guess, to go through the environmental factors that influence bacterial growth. Temperature, oxygen, salt content, which is going to be related to the activity of water, pH, and radiation. So all of these things can limit bacterial growth. And understanding how they work allows you to, try and keep, for example, different food types and protect them from bacterial contamination. Okay, so for the effect of temperature on bacterial growth, for any kind of bacterial strain, you have three cardinal temperatures. You'll have the minimum temperature below which bacteria will not grow. You'll have their optimal growth temperature, and then the maximum temperature over which they won't grow. So you have some kind of relationship. As the temperature increases, then you'll have, as the temperature increases, the growth rate will reach a peak, and then as you increase the temperature, then growth rate will go down and bacteria won't grow. So that's because, at higher temperatures, proteins will denature, membranes won't function anymore, and at lower temperatures, well, enzymes won't be working very effectively. They require an optimal temperature. And also membranes, they need to have a particular fluidity. If the temperature is too low, membranes won't function anymore. Now, just a quick question here. So when we talk about the growth speed or growth rate, which is going to be correlated with growth speed? Don't click on this. We'll just do this one by a show of hands because we have to move a bit quickly, I think. So first think about it. Which of them is directly correlated to bacterial growth speed? Doubling time G or the inverse? Okay, everybody decided? Who says the growth speed is directly correlated to the doubling time? Nobody. Who says the growth speed is inversely correlated to doubling time? Yeah, you're right. So that basically this just means that the bigger the doubling time is, the slower the bacteria grow. Okay, the shorter your doubling time, the faster they're growing. So this curve here, right, this value here, this is 1 over the growth time. The generation time, rather, not growth time. Okay, so depending on the cardinal temperatures for bacterial growth, there are these different categories of bacteria which tell you something about their relationship with temperature for growth. So most bacteria that we study in the lab anyway are going to, or in TP, are going to be mesophiles. That means that their optimal growth temperature is between 20 to 40 degrees centigrade. They won't grow above 50, and they won't grow below 15 degrees. Now, some bacteria, so Escherichia coli, Staphylococcus aureus, they're mesophiles. Some bacteria, like Listeria monocytogenes, are what's called psychotrophic. So these bacteria will still have a growth optimum about 20 to 30 degrees. They won't grow above 35, or they won't grow above 40 maybe, but they can still carry on growing down to 0. So that can be important because Listeria, they're very famous for contaminating like meat products, like pate, stuff like this, and they can carry on growing in the fridge. So, you might have kept it in the fridge for 2 weeks. If you've got Listeria contamination, the bacteria can really, really grow and make you sick afterwards. Psychrophile, these bacteria are highly dependent on low temperatures. Okay, you get a bit psychrophilic bacteria in very cold environments in the Arctic, in the Antarctic. So their optimal growth temperature will be 10 degrees centigrade or less. They won't grow at temperatures above 20 or 25 degrees, and they can grow down to temperatures of -5, especially if they're growing in salt water, okay, because salt water will not freeze at 0. Thermophiles, okay, these thermophiles will only grow above 45 degrees. Growth optimum 55 to 60, but they won't grow above 80 degrees. And the bacteria that grow above 80 degrees are hyperthermophiles. So Geobacillus stereothermophilus is a thermophile, Thermus aquaticus thermophile, and these extreme hyperthermophiles will be growing in volcanic vents down under the ocean, which are under high pressure. So the temperature of the water can actually be above 100 degrees because the pressure is so high. So some of these guys can grow there. So there are a number of molecular adaptations to growing at high and low temperature. So for example, the membrane lipids in thermophiles and hyperthermophiles will have longer chains of fatty acids, so that the melting temperature of the lipid is much higher, okay? And it will be the opposite for secrophils. So their membranes will be much more fluid at room temperature, and even like they won't function anymore if they're at room temperature of 37 degrees. Similarly with proteins, the proteins of thermophiles will have a number of adaptations. They'll have perhaps a higher number of cysteine bridges, okay, and they will also have a lot of buried hydrophobic amino acids. Okay, so the hydrophobic interactions become stronger with temperature, and these kind of allow these bacterial proteins to remain in a native state even at high temperature. So these are very important industrially. If you want to get enzymes that work at high temperatures, you extract them from thermophiles and hyperthermophiles. If you want to get enzymes that will work outdoors in a low temperature, you can get them from psychrophilic or psychotrophic microbes. Because for mesophile, if you want to use their enzymes outside, most of the time they're not going to work so well, except in summer, Okay, oxygen relationships. So this is a huge classic. Everybody needs to understand this one. So what you can do, and what we will do experimentally, is inoculate bacteria down into a long tube of agar with a nutritive medium, and then see where they grow. And what happens is that if you leave the cap unscrewed, then the top of the tube will be open to oxygen, whereas down in the base of the tube you have no oxygen. So you get a kind of gradient through the tube. So down at the base, no oxygen. Up at the top, you have 20% oxygen, just like in the atmosphere. So bacteria that only grow at the top of the tube are obligate aerobes. Bacteria that grow better in the presence of oxygen, but can grow all the way down, are called facultative anaerobes. Bacteria that grow all the way through the tube, but they don't actually use oxygen, okay, so the density is going to be the same all the way through, are called aerotolerant anaerobes. And bacteria that only grow in the bottom are called strict anaerobes, so they cannot survive the presence of oxygen. And there are also some bacteria which are microaerophilic, which means that they require oxygen to grow, but they can't survive high oxygen concentration. Now, there are a few examples of the bacteria in different categories here. So E. coli, Escherichia coli is a facultative anaerobe. The genera Bacteroides or Clostridium, classical strict anaerobes. Obligate aerobes, most Bacillus are, some Bacillus subtilis is considered to be a obligate aerobe. Some other Bacillus can be anaerobes. Mycobacterium are obligate anaerobes. Now, the reason some bacteria can survive oxygen and others can't is because of the presence of these two enzymes which detoxify reactive oxygen species. So, strict anaerobes do not express either of these enzymes. So SOD is superoxide dismutase. And the reaction catalyzed by this enzyme is to take an oxygen radical, okay, so this is a molecule of oxygen, O2, which has got one extra electron. So it's a superoxide radical. This is very, very reactive. It will react with DNA, react with nucleic acids. It's toxic. and add that to, no, that's wrong, isn't it? 2 protons, and that goes to a molecule of hydrogen peroxide. Now, this, yes, this one. I'm not surprised. Is that better? Okay. Okay, so this is a superoxide and it's being catalyzed to hydrogen peroxide by adding 2 protons. So this is very toxic oxygen molecule, but hydrogen peroxide is also toxic. Okay, you can use it to disinfect a wound, for example. Now, strict anaerobes do not possess either of these enzymes, so they're very sensitive to the presence of oxygen. Now, aerotolerant anaerobes will only have superoxide dismutase, but no catalase. Catalase is what? Catalase is going to catalyze the splitting of hydrogen peroxide into water and a molecule of oxygen. So if you have these 2 enzymes, then this superoxide radical can be completely converted into harmless compounds. Okay? Whereas if you only have the superoxide dismutase, you can reduce the toxicity of the superoxide radical, but you still have a toxic byproduct. Now, for bacteria that do not perform aerobic respiration, okay, they are not using oxygen in their metabolism. That is aerotolerant anaerobes. These guys do not use oxygen. Then just having superoxide dismutase is enough to protect them. Whereas these guys, obligate aerobes and facultative anaerobes, when they grow in the presence of oxygen, they are using oxygen, as we'll see in subsequent lectures, well, you will see anyway. Oxygen is used as the terminal electron acceptor in respiration. So what is happening is that during this respiration, you can get an increased production of this superoxide radical because of the metabolism of the bacteria. And in order to and if they only have superoxide dismutase, these bacteria will accumulate a concentration of hydrogen peroxide that is going to be too high and will be toxic. So for bacteria that use oxygen in their metabolism, in respiration, obligate aerobes and facultative anaerobes, their metabolism is going to be producing a bigger concentration of this superoxide radical. And they need both of these enzymes in order to be able to survive. Okay, the situation with microaerophilic bacteria, they all definitely have superoxide dismutase. Some of them have catalase, some don't. Okay, oxygen relationships, very important. This one comes up in exams and CC and CC very frequently. Okay, so one of the things you wonder about is how can you actually culture strict anaerobes, and especially how do you isolate them on an agar plate, because a lot of bacteriology depends on isolating one colony that you can then study. And well, there is a little trick that allows you to grow bacteria on agar plates, even, well, grow strict anaerobes on agar plates. So what you do is you put them in this kind of jar, and here's all the agar plates stacked up. You have a kind of like sachet full of some kind of chemicals in there, and you add water and then close the lid of this jar, and all the oxygen is going to be used up. So the chemicals you have here are a mixture of sodium borohydride, citric acid and sodium bicarbonate. And then when you add water to this, what's going to happen is you'll produce hydrogen gas from both of these reactions. And then you have a palladium catalyst somewhere up in the lid here, so the hydrogen will react with the oxygen to produce water, and this will remove all the oxygen from the atmosphere inside this jar. And you also have agar with a special additive to absorb any residual oxygen. And you've got like a little kind of strip here which will turn white, just to show you that your system is working. So this allows you to, work with anaerobes and just grow them by having this special jar where you can remove all the oxygen. And this allows strict anaerobes to grow. In liquid culture, it's not so problematic. They'll just grow down in the bottom. Okay, you can degas the liquid, inoculate the bacteria, and they will grow. But to get colonies, you need some kind of special setup, which is like this. Okay, so we've seen temperature, we've seen oxygen. Now another part of the chemical environment that influences bacterial growth is pH. Okay, so most bacteria prefer to grow around about neutral pH, in between pH 5.5 and pH 8. So these bacteria are called neutrophiles. Acidophiles grow at acidic pH. Now, the pH optimum really depends on the particular strain of bacterium. Some bacteria will have a pH optimum of about 4. Others, extreme acidophiles will prefer to grow at pH 2. Okay, so you have things like Sulfolobus, Ferroplasma. Blue, whenever I've got blue here, that means it's an archibacterium. Okay, so similarly for this one, Pyrolobus, hyperthermophile, it's an archibacterium. So you have acidophiles, and you also have bacteria that prefer alkaline environments. So you can find them in alkaline lakes, for example. So Bacillus alkalophilus. Somebody's kind of not really very inspired for a name here, right? But helpful. So, they prefer alkaline pH, but they can't really grow above pH 10. Now, what is kind of interesting here is that the cytoplasm of the bacterium will stay close to neutral, even in bacteria that are acidophiles or alkalophiles. So there's some kind of homeostasis that bacteria perform to be able to grow in acidic conditions. So for example, Escherichia coli, it's a neutrophile, but if its conditions are acidic, what it will do is it will use a proton ATPase to pump out protons from the cytoplasm outside the cell so that the pH inside the cytoplasm can stay close to neutral. And alkalophiles will do the opposite. Okay, so they'll try and accumulate protons inside the cytoplasm to resist the alkaline pH outside the cell. And they can do this sometimes by exchanging sodium, which will be exported, in order to import protons. So you have some kind of like pH homeostasis in bacterial cells. Now, another thing that's kind of similar is the relationship to bacteria with salt concentration. So once again, most bacteria pretty much prefer to grow at salt concentrations lower than 1 molar. Okay, around about 0.2 molar will be their optimum. So there's no real word for this. You just call them non-halophilic bacteria. Just to give you some context, so Serum concentration of salt is about what in the human body or other mammals? Anybody know? What's the matter? Don't you hate physiology or what? Nobody have any guess? Like we got 0.2 molar here. Who says salt concentration in serum is higher than that? Who says it's lower? Yeah, you're right. It's a bit lower, isn't it? Yeah. So 142 millimolar, 150 millimolar salt is isotonic for human cells or mammal cells. Seawater, the salt concentration about 0.6 molar. Okay, so more than this. So most eubacteria will grow in isotonic salt conditions, the same as like a eukaryotic cell, mammalian cells, and will probably also grow in seawater. Okay, 0.6 molar. Now bacteria that can grow at concentrations of salt higher than 1 molar are halotolerant or halophilic. So for example, Staphylococcus is halotolerant, as we saw in the Chapman-Agar. Differential and selective for Staphylococcus, differential for Staphylococcus aureus. So in Chapman-Agar, you've got 75 grams per liter of salt, which turns out to be about 1.25 molar. Okay, so this is a high enough salt concentration to prevent the growth of most eubacteria, because most won't grow above 1 molar salt. So it's selective for halotolerant or halophilic bacteria. So halotolerant bacteria, their optimum salt concentration will be about the same as non-halophiles, but they can resist and they can grow in salt concentrations above 1 molar. So halophilic bacteria, they require salt concentrations higher than 1 molar, okay? And they won't grow at salt concentrations less than 0.5 molar. So the example I found for halophile is Ectothiorhodospira, which probably no one has heard of, but you might have seen it because they are purple photosynthetic bacteria that grow in high salt environments. Can you imagine where that might be? Somewhere near Nantes? Slightly purple color, high salt. Actually, I have some here. Oh, he's gone. Oh. Celle de Guérande. So, in the salt marshes, you've got a very, very high salt concentration. If you go there and look, you see it's kind of got a purple color. So that's probably these bacteria growing in there, okay? And they can survive salt concentration, above 2 molar salt. Really quite impressive. And then you have like some extreme halophiles that can grow up to 5 molar salt, like halobacteria. Okay, so how do bacteria survive these salt concentrations? Because once again, there's homeostatic control to try and maintain a constant salt concentration in the cytoplasm of the bacterium, or at least retain water, okay? Because the problem for bacteria is that if the salt concentration is too high outside, then all the water content of the bacterium is going to be too low, because the water will tend to diffuse out into this high salt concentration. If outside the bacterium is hypertonic, then the salt concentration is low, the opposite will happen. The water will diffuse into the bacterium and could create osmotic pressure. So bacteria have got to control their internal environment with some kind of response. So in a hypertonic solution with high salt concentration outside the bacteria want to retain water, okay? And they do that by accumulating the concentration of, increasing the concentration of what are called compatible solutes. So these can be free amino acids, okay, proline, glutamate. So amino acids, they have a charge. They've got a positive charge and a negative charge. Because they've got a charge, that means they've got a kind of osmotic, well, they increase the osmotic pressure, but they don't change the pH, okay? And they can also increase the concentration of these glycine betaine solutes. And glycine betaine is something like this, okay? So it's got, once again, positive charge and negative charge. It helps to retain water. Now, another example, so most bacteria will do this, you know, like Escherichia coli, Staphylococcus aureus will be doing this. Extreme halophiles like Halobacterium, they'll accumulate potassium to try and like balance out the sodium that's really high concentration outside the cell. Now, if bacteria are in a hypertonic medium, it's the kind of the opposite. So these solutes, they won't be free in solution anymore. They'll polymerize into inclusion bodies. So their free concentration is much lower, and that means that they won't retain water so much. Okay. And the other thing is that some of these solutes will be released outside the cell by pores which are mechanosensitive. So if the osmotic pressure is too high inside the bacterial cell, it's going to like explode the cell if it carries on, or put too much pressure on the peptidoglycan, then these mechanosensitive pores will open and release some solutes. So there's some homeostatic resistance to salt concentration. Now, salt concentration is kind of related to something else called the activity of water. And the activity of water is just the concept that, you know, if you've got some kind of substrate with a certain amount of water molecules in it, not all of the water will actually be available to interact in chemical reactions. And so if the water in the substrate is less available, it can't really evaporate so much from whatever it is. And so this is kind of gives you an easy way to measure the activity of water in any kind of substrate. Like it can be liquid, can be a foodstuff, and so on. And it's basically 1/100th of the relative humidity that is released from this substance, whatever it is. Okay, so if you've got something like a bucket of water and you allow it to equilibrate in a closed chamber, the relative humidity above that water will be 100%. Okay, everybody understand this? Okay, you've got a closed chamber like this. You've got something here with pure water. And it's going to equilibrate with some water vapor in the gas phase. And this will be, once it equilibrates, this will be 100% relative humidity. Because at the equilibrium, it can't get any more. So that's your 100% relative humidity. But if you've got something like, I don't know, something, and it's not actually pure water, it's something like honey with a lot of sugar in it, okay? A lot of the water is not going to be evaporated because it's like interacting with the sugar molecules in the honey. So this will release less relative humidity. And that tells you what the activity of water is. So I'm going to skip this question. We can try this one other time later on. Now, people who do food microbiology, they spend their lives measuring the activity of water in different types of foodstuff. Okay, that's probably a bit of an exaggeration, but it's useful because once you do this, you realize that, you know, most fresh food, you know, fresh meat, vegetables, fruit, well, blood, you know, something like a serum, the activity of water in these fresh substances is greater than 0.98. So it's close to what you have just with free water. And that means that pretty much all bacteria, gram-positives and gram-negatives, will be able to grow on these substances. Okay, so there's no limitation. Something like bread, activity of water is a bit lower. Ham, the activity of water is lower. So less than 0.95, gram-negative bacteria won't really grow. Only some gram-positives. Once you get down to an activity of water about 0.85, you'll only be able to have staphylococcus growing on this tsunami. And then once you get down to an activity of water of 0.8 or 0.7, then really no bacteria will grow on this type of foodstuff. What kind of foodstuff am I talking about? Things like cereals. I don't mean cornflakes, Rice Krispies and stuff like that. I mean wheat, rice, that you buy in a pack or something like that. So once you've got a nice bit of dry, grain of wheat or a grain of rice, bacterial contamination is not a problem because there is not enough free water in these cereals for the bacteria to grow. The real problem comes from fungi. Okay, these are Penicillium and Aspergillus, which can contaminate cereals. This is why if it's raining around harvest time, that's bad for everybody because the grains will have a higher water content and you can easily get a fungal contamination, which can spoil the crop. So this means that, depending on what the activity of water is, then you can control what kind of contamination you might have inside the foodstuff. Now, you can exploit these things together, pH, oxygen, and activity of water, to conserve food. So, for example, if you go buy some pita bread like this down at your local supermarket, if you look at the expiry date, they can last quite a long time, okay? And they won't get contaminated. Now that's because it's a little bit dry, activity of water 0.95, so gram-negatives won't grow, some gram-positives might grow. Fungi can grow on this, molds can grow on it, but actually the atmosphere underneath here has no oxygen. And all molds, fungi, Aspergillus penicillium, they are obligate aerobes. So you can't get any growth because there's no oxygen and the activity of water is a little bit too low for most bacteria. So often, if you see some kind of like penicillium, a little green spot here, what's happened is the pack has been damaged and you've got oxygen that's got in there. And then the spores that are already present will germinate and begin to grow. Now, this one, another example of fantastic long conservation food product. Anybody know what this is? Has anybody eaten any of this? Come on, admit it. You're really hungry, right? 3 a.m., anything looks good, right? Okay, dried dog food or cat food, these kind of pellets. The activity of water here is really low, 0.65. So nothing, no fungus, no bacteria can grow on this. As long as it stays dry, then it can keep for months, okay? And then your dog can just come and eat it, and you don't need a fridge, you don't need special conditions to keep this food. Now, something we've probably all eaten, some hard cheese like this. If you keep this down in a cellar somewhere, it can last for 1 year, 18 months, 24 months. It even gets more expensive the longer you keep it. That's because the pH inside is very low. Most bacteria won't grow, and the activity of water is quite low. So fungi can grow on this, but they'll grow on the outside, and that's what the rind is, okay? So this is protecting the food from getting colonized by anything else. And last thing is something like this, yogurt, pH is going to be low, so most bacteria won't grow, most fungi won't grow, and of course, it's conserved at low temperature. So as long as the temperature, you keep this in the fridge, it could last quite a long time, And I have to admit, I've eaten yogurts that are way past their expiry date, and they're perfectly fine. Well, at least I'm still alive anyway. Okay, we just have a couple of things to finish, which I'll try to do in 5 minutes. We just have like radiation, basically, and that's it. Okay. So radiation, different types of radiation, ionizing radiation. Infrared, visible light, they are not dangerous, not harmful to bacteria, but ultraviolet radiation can have an impact on biological systems. So ionizing radiation, alpha, beta, and gamma rays. they're mainly toxic because as these rays traverse or go through or penetrate a bacterial cell, then they'll interact with the water molecules that are there and produce reactive oxygen species. So that's why radiation is toxic for bacteria and for us, because these reactive oxygen species will then go on and interact with, react with DNA and damage it chemically. So there are some bacteria that are highly resistant to radiation, like Deinococcus radiodurans can survive a huge dose of radiation, but it doesn't actually live near any source of radioactivity. And the reason that they are so tolerant of radioactivity is because they have a lot of enzymes to DNA, to repair damaged DNA, and they also have enzymes to detoxify reactive oxygen species. So it's a kind of like crossover of their like resistance to oxygen, which allows them to also resist radiation. Now, ionizing radiation is not that frequent in the natural environment, but it's very useful for the sterilization of, things used in the laboratory, or like the plastics that we use in the lab. They don't resist high temperature, so they're all sterilized by radiation. And also solutions of injectable drugs. You can irradiate them and you can be sure there's no bacteria in there. Okay, for UV, 3 different types of UV, A, B, and C. And as you get further away from visible light, you go from A to C. So A has got a longer wavelength than B, and B has got a longer wavelength than C. So of course, the energy you have in the radiation, in the light ray, is higher with a shorter wavelength, right? So UVC is going to be the most toxic. Now, UVC is highly toxic, absorbed by DNA. It's absolutely lethal for everybody. The only reason we can live on the planet of the Earth, on the surface of planet Earth, is because UVC is absorbed by the ozone layer. And until the ozone layer had formed around the planet, the surface was absolutely uninhabitable because of too much UVC. Now, this is UVC radiation is also used in some kind of procedures for sterilizing water. Maybe not in a swimming pool. Also, I think I've seen UVC irradiation used in airport security during COVID-19. They would like all the stuff that you had would go through a UVC thing, and that's enough to like kill bacteria and or any viruses. So it had some kind of use, and of course it's not so dangerous as ionizing radiation. UVA and UVB. Okay, toxic, well, UVA, not so harmful, but still can have some biological effects. UVB is the one that is dangerous, can cause cancer, okay, if you stay in the sun too long. And it's absorbed by DNA, but it's also got an indirect effect of reactive oxygen species. Okay, so, because of this, there's some kind of crossover between resistance to oxygen and resistance to radiation. Okay, right, so that's about it, I think. So I'll just finish up here. We're about on time. So the environmental factors that influence bacterial growth: temperature, availability of oxygen, pH, salt content, the activity of water, and radiation. So all of these are important, and you have to know the terms: halophilic, halotolerant, acidophile, and so on. So, and it would be nice if you knew one example of a bacterium for each kind of category. That's why I put them in the slides. Okay, well, thank you very much for being here today, everybody, and I'll see you, well, later, much later. Bon, allez. Tomorrow morning.